will share the results. So these are our motivations. We already knew that arterial function was regulated by ion channels present in their smooth muscle cells. Additionally, our preliminary experimental data showed that smooth muscle cells' ability to contract changed nonlinearly with disease progression. Based on that, we hypothesized that hypertension nonlinearly changes L-type calcium current kinetics. Therefore, this project's long-term goal is to quantify hypertension-induced biophysical changes in the L-type calcium current. Now, I've been talking a lot about the L-type calcium channel and its current, but what are they? Well, this is a diagram of a cell, and this is the L-type calcium channel. Now, this equation up here at the top right can be used to model the L-type calcium current. AM here, this is the surface area. PVOCC is the conductance. DL here is the activation, which is also written here. FL is the inactivation, which is written down here. And of course, this equation is dependent on CAO and CAI. So the activation is made up of the steady state of activation, which is written here first, and the time constant of activation, which is written second. The same is true for the inactivation. Here are some graphs that show the activation and inactivation on the bottom left. Now, the most important thing is that these parameters here, highlighted in purple, are all unknowns and have been values that I've been trying to estimate under healthy and hypertension conditions. I will talk more about those parameters later, but first I will tell you about the experimental data we obtained from Roberts. The data was collected from rat neurons using a voltage clamp potential. With this method, a pipette was used to control the transmembrane voltage while measuring the current flow through the L-type calcium channel. The data gave us an initial graph that looked like this. Now, here's the graph data for the control cells, and here's the graph data for the hypertension. As you can see here, we graphed the L-type calcium currents against time, the activation against time, and the inactivation against time. From the collected data, we could initially conclude that hypertension increases the L-type calcium current. This is a graph of the clamp potential graphed against the peak L-type calcium current, and here is the same graph for hypertension. As you can see here, for the control graph, the peak current was about -47 picoamps, and for the hypertension, the peak current was about -130 picoamps. We therefore concluded that the hypertension increased the L-type calcium current. After making that initial conclusion, we decided to try and estimate the values I had previously mentioned by graphing our recorded data and comparing it to other graphs made with our estimated values. So we modeled the L-type calcium channel using the Goldman-Hodgkin-Katz model. And the equation for this model is the one I showed a couple slides ago. We also estimated our values using a previously established Nelder-Mead algorithm, which Dr. K talked about in his presentation yesterday. I also assisted in implementing a new simulated annealing method, which simply put is trying to make a guess and then more guesses and seeing if those are better. We also used trivially parallel serial farming, which basically means that instead of spending maybe 10 minutes, which is what I had to spend trying out each set of parameters, we had a computer run a bunch of sets of parameters at a time, and then I was able to look at the results afterwards. So my results, what came out of this? Well, as I mentioned before, hypertension increases the peak L-type calcium current. We also found that our preliminary fitting results showed that hypertension shifts the L-type calcium current steady state activity to lower membrane potentials, which is indicative of increased intercellular calcium concentration or increased hypertension. At the moment, that's everything I found, but in the future, I intend to develop a new smooth muscle cell model capable of producing experimentally verifiable hypotheses and reproducing hypertension-related data. That's the end of my presentation. Are there any questions?
So Prof. Bisbee has a question, Jay.
Yeah.
Yes.
Well, I haven't typed it out, sorry.
Oh, it's okay.
So does this matter at all what model of hypertension we're dealing with, or is it just hypertension in general?
Oh, these are model. Yeah, yes, it matters. So we're modeling, this is a model based on rat neurons right now. So we're hoping to model neurons.
Rat arterioles.
Arterioles. Mm-hmm.
Did you just yell that down the hall at?
Yeah,
okay. Okay, so this doesn't, so what model of hypertension is this?
Oh, this is the, sorry, here, I can show here. Sorry, this is the Goldman-Hodgkin-Katz model that we're using.
That's just a model of ion behavior.
To
Yes.
add to what Jay has just said, Prof, this is a model of hypertension where the lumen is put under high pressure. So you fill the lumen of the arteriole with lots of liquid.
Oh, so this isn't from
high pressure.
These aren't hypertensive animals? Okay, so you go in there, you haul out the blood vessels, and then you dissociate the vascular smooth muscle cells, and then you put a patch on them, right?
Something like that.
What rat, I'm assuming they're rats, what rat model are you using?
No idea. No idea.
Is it even a hypertensive rat model? Because overinflating or increasing intravascular pressure on a blood vessel that day is not hypertension. So
I apologize for my ignorance, but I will find out.
Okay, so this is all I want to say is hypertension is a chronic disease. Okay, and you know you can have we've already talked about salt-sensitive hypertension with I think it was Riva's talk. Was it Riva's talk? I think so. So we talked about salt-sensitive hypertension. There's neurogenic. There's renal insufficiency. There's a zillion different reasons why people get hypertensive, and there's also at least that many reasons why animals get hypertensive. I mean, I can pull a renin gene right out of a rat, and it's going to get hypertensive fast. But that is a chronic condition where the vessel adapts. Okay, so blood pressure goes up, you have autoregulation in the brain, you have all of the neural, endocrine, renal mechanisms that go into regulating blood pressure, but you still have a hypertensive phenotype. Okay, but that is something that is a chronic day in, day out thing because the system is somehow not functional or not appropriately functional. So we'll give people an ACE inhibitor and they'll, pee out extra volume and and things like that. And that's all fine. But be careful the terms you use. This is an acute increase in intravascular pressure. And by by calling it hypertension, though, you're going to have people are going to think something very different. So I was thinking entirely about a hypertensive rat model, like a dull salt sensitive rat as an example. And then, you know, what causes that animal's vascular smooth muscle in the brain to change to adapt to the hypertension? Because what kills these animals almost invariably is a stroke.
Jeff, I believe the experimental data is for a specific process that happens with collections of these proteins, these L-type calcium channels.
Yep. Oh, I get it. I'm just curious where you got the vessel, where you got, you know, where you got the tissues.
And we decided, I decided to call it hypertension with J, just to keep language simple.
Okay. Well, you can just call it high pressure. I mean, that's fine. I mean, hypertension is a clinical thing that causes people's minds to spin off into other. It's like calling it diabetes just because you added glucose to the medium. Okay, not diabetes.
Absolutely.
So, but anyway,
this was us keeping the language simple.
Yeah, that's fine.
And accessible.
Well, that's why we're talking.
But I hope you have time to talk about the process that Jay has been referring to.
I absolutely do, and because I don't want to slow down the day any more than I already have. So so I'll back off a little bit. But
I mean, we can talk about this in depth on the time.
Yeah, we can talk about L-type calcium channels, what they do, why, and then, you know, why the vessel doesn't just do this and turn into nothing.
Yep.
Like there's a reason why. So, okay, thank you. I'm good.
Thank you, sir. Thanks to Prof. Frisbee and all attendees for making our first full session exceedingly successful. Congratulations to all. And I now propose that we break for lunch till 2 o'clock. So for